Hi friends, today we are going to see pharmaceutical process development, how the product is developed in the pharmaceutical industry. Mostly in pharmaceutical industry, when any product would like to develop, it is decided for the development and the dosage form to be fixed. So pharmaceutical process development is, we can say that a vast subject, but we will see one by one as per the definition. Process development refers to the systematic approach used to the define how a drug substance means API, active pharmaceutical ingredient, and drug product formulation will be manufactured from laboratory scale to commercial production. Means what happened in pharmaceutical industry? First, we have to take a small batch, very small batch, that is called as the scale-up batch. And basis on that scale-up batch, all the study, all the results, we are going to the commercial. But so how this small batch is developed, how it will be decided, we will see step by step. But the intention is that product should be considered or ensured for the quality, safety and efficacy. Means it should have quality, it should be safe for the patient, and it should have efficacy of the medicines. Why process development is required? If suppose we want to say to meet the patient requirement, to understand the product nature, to achieve the efficiency and cost reduction, innovation and growth. Suppose today we have received any of the demand from market that this product is required, or sometimes demand is not coming, but company as a company decides this molecule we have to decide. New research is going on, new product is decided, and based on that molecule they are deciding to run the product. So in that scenario, what they do, they will first see the development mechanism with respect to patient safety and the product nature and other things, as well as the innovation and growth, because some innovations consistently going on for the development and the growth part of company. The key objective of this product development is to meet the patient expectations, market requirement, and develop the robust and reproducible manufacturing process. It also ensures the product quality should meet the regulatory standard. In the beginning, the product should be taken on very small batch, like laboratory level or pilot batch, scale up batch. We can say that in the small level, and to design the critical process parameter, critical quality attributes, product is designed. Also, it should have cost effectiveness and user friendly. Means what will be the dosage form, like tablet, capsules, liquid. It say that user friendly, and the quality and sustainability of the product should be there. First, to identify the dosage form to produce, means which product we have to run or which product we have to develop. In which dosage form they are decided, like tablet, liquid, injectable, whatever is there. Basis on that, they select the key element, raw materials based on the prior experience, design the formulations with respect to dosage form, like tablet, capsules, liquids or injectable, whatever is required, identify the critical quality attributes and critical process parameters. Critical quality attributes means those are the critical part of the product, like solubility, dissolution, assay, means all the parameters relevant to the product, that is critical quality attributes, and related substance, that is impurity profiles, all this relevant to the product profile. Tablet hardness, in case of liquid, pH, weight per ml, all comes into the quality attributes. In this scenario, they first decide the master formula to prepare the product, select the key raw material, basis on that solubility, product profile, material nature, all these ingredients they decided, and decide weight per ml per tablet. means how much is the weight of each ingredient should be in that tablet, it is decided. Tentative limit for hardness, thickness, diameter, weight of the tablet, all this decided. Average weight of the tablet with plus minus limits as per the pharmacopoeia, whatever the limit is there. Water quantity and choice of binder, nature, may paste preparation with starch or water. So whatever the binder is required, accordingly they decides. Later on, with respect to equipment selection, they select the equipment like a sifter for sifting process, sifting of ingredient, mesh size, which mesh to be take for the sifting of material, multi-mill if suppose milling is required, rapid mixer granulator for the mixing of the granules. All the granules, all the material, raw material will be added into the rapid mixer granulator for the proper mixing. Then fluid bed dryer, and after that blender. All these equipment they selects. Compression machine and capsule filling machine as required of the dosage form. Suppose if you want to make the tablet, we will use compression machine. If you want to make the capsules, we will select the capsule machine. And suppose some of the times if tablet requires coating, that time coating machine to be selected. So initially small scale of batch to be planned. As per requirement, all the raw material and machine availability to be arranged. Batch size will be decided. In the beginning, for scale up batch, very small size batch sizes are there, like 5 kg, 10 kg batch sizes will be there. Batch size will be depend on equipment capacity of lab scale and pilot scale. Sometimes, suppose bigger equipment in the pilot area or formulation and development area, that time 1 lakh batch size or 100 kg batch size also they can plan. But mostly starts from beginning 5 kg or 10 kg batch sizes. Knowledge and understanding of its physical, chemical, biological and microbiological properties of characteristics which can influence development of the drug product and solubility of the drug substance can affect the choice of dosage form. So all the chemical material, all the materials to be verified for the chemical and physical properties, because most of the time chemical and physical properties impacts on the product nature. Total quality target product profile to be designed and potential CQAs of the drug product defined. So the quality target profile, QTPP and potential CQAs of the drug product as defined in ICH can help to identify and on development and manufacture of the drug substance. Quality risk management, QRM describe. This can be used a variety of. So product development approach. In this scenario, strategies for product development may vary from company to company and from product to product. The approach or the extent of the development can also vary and should be outlined in the submission. Whenever we are going to submit the product in the dossier, that time we have to take this care. And most of the thing, product development is vary from company to company. These concepts apply equally to the development of the drug substance manufacturing process. A systematic evaluation, understanding and refining of the manufacturing process, including identifying thorough, easy prior knowledge, experimentation and risk assessment. The material attributes and process parameter that can have an effect on the drug substance CQAs. Drug substance CQAs typically include those properties or characteristics that affect identity, purity of the drug, biological activity, stability. When the physical properties are important with respect to in vivo performance or drug product manufacturing, this can be designed as a CQA. Means most of the time we are doing in the initial phase in vitro study, vivo study, because for the dissolution or some product characteristic, whether the product is properly dissolved, soluble or effect or efficacy to the patient, that type of study is going on. So that time we are designing the CQAs. In the case of biotechnological or biological product, most of the CQAs of the drug product are associated with the drug substance. Here, most of the time, it is associated with drug substance. Thus, are a direct result of the design of the drug substance and its manufacturing process. Determining the functional relationship that link material attribute and process parameter to the drug substance. Most of the time, whatever the attributes of the raw material, it will impact on the final finished products characteristic or the drug substance CQA. Drug substance CQA typically include those properties or characteristics that affect identity, purity, biological activity and stability. Means what we are seeing here, critical quality attributes typically includes or characteristics that affect the identity, purity, biological activity and stability of the product. If suppose hardness of the product is not good, if suppose assay of the product is not coming into the results, so or impurity is more, so ultimately it will be impacting on the product stability as well as the purity. When physical properties are important with respect to in vivo performance of drug product manufacture, this can be designated as CQA. In the case of biotechnological or biological products, most of the CQAs of the drug product are associated with the drug substance, and thus these are direct result of the designing of its manufacturing process. Material attributes and process parameters to the drug substance, that is CQAs, the manufacturing process development program should identify which material attributes, raw materials, starting materials, reagents, solvents, processes, intermediates, and process parameters should be controlled. What we have to control? It should be given in the CPP. Like as most of the time CQA impacted because of CPP. CPP means critical process parameter. Suppose process parameter means the product heating temperature, timing, then mixing timing, all these suppose there is a limits for the length between two process, okay, or timing. So it depends on the CQA, means it will impact on the CQA. Suppose there is a standard minute time, 30 minutes drying to be done. But suppose in case or by mistake 40-minute drying is happened, it will directly impact on the product CQA. CQA means product assay or impurity or dissolution. So the strict process parameters should be followed. Whatever suppose during the designing phase we have decided like 30-minute drying, 20-minute drying, that should be strictly followed and recorded. Risk assessment can help identify the material attributes, process parameters with the potential for having an effect on the drug substance CQAs. When assessing the link between an impurity in a raw material or intermediate drug substance CQAs, the ability of the drug substance manufacturing process to remove the impurity should be considered in the assessment. Means what assessment we should do? During the raw material testing or intermediate, we have to remove most of the impurity. If suppose in the raw material itself the impurity is on higher side, suppose limit is given as not more than 0.1%, okay, and you got around 0.08, means around to 0.01. So in that scenario, the same impurity will increase into the finished product or drug substance. Means in the simple way, raw material should be stringent and very much specified, and the impurity should be very less in the raw material. If on the border side or higher side in the raw material, it goes into the finished product on the higher side as well. Identify the potential sources of process variability, identify the material attributes, process parameters likely to have greatest impact on the drug substance. Means process parameter, like earlier we discussed, temperature, heat, timing, all these impact on the drug substance quality. This can be based on the prior knowledge and risk assessment tools, design and conduct experiment, or mechanistic studies, means multivariate design of experiments, simulation, modeling, Means during study, during product development, we have to make different type of study, like 30-minute drying, 20-minute drying, 10-minute drying, and whatever the fits or good giving the results, that limit fix into the BMR or the process. So analysis and assessment of the data to establish appropriate ranges, including the establishment of design space if desired. Means we have to make the design space during the beginning phase or during the pilot scale up batches or scale up batches. So how it means by design space? Suppose design space means your limit is drying limit is 20 minutes to 40 minutes. So this is the design space. In commercial or during the next batches, you can reduce that limit. Rather than 20 to 40, you can make it 25 to 35. Means in that design space, you can work, and based on the product result, some stringent control, you can reduce that limit. Design space is the ability to accurately assess the significance and effect of the variability of material attributes, process parameters on drug substance, critical quality attributes, and hence the limit of design space depends on the extent of process and product understanding. Why these limits is given wider? Because of depend on the process and product nature, understanding of the process and product nature. So the limits are given wider, like 20 to 40 or 50. And when the product is continuously monitoring, continuously manufacturing, over a period, this limit can be made stringent, like 25 to 30. It is important to understand the formation, whether the impurities react and changes to the chemical structure, purge, as well as the relationship to the resulting impurities at the end of the drug substance and impacted on the CQS. Means suppose in your raw material has the impurity on higher side, or during the API manufacturing, if impurity is there in the API itself, so that same impurity can increase into your finished goods, drugs, or the drug substance. Process development information should be logically organized, easy to understand. Manufacturer can present the process development formation in a number of different ways, but some specific recommendations are provided below for the considerations. Means your process development should be very logical, like first sifting of the material, then mixing of the material, then binder addition, then mixing of the material, then material goes to the FBD, like fluidization, drying of the material with some heating condition. And when that process is completed, then material will be unloaded. Then again it will be milling if required for the proper granules, then again sifting, and then go for the blending, and then compression. So that type of logical sequence should be mentioned in that particular product development. Brief description of the stages in the evaluation of the manufacturing process and control strategy. Brief description of material attribute. Material attribute should have a brief description. Means suppose particle size, what will be particle size, what will be the color of the material, what will be the granular size, what will be the solubility. All these characteristics are the attributes of the material, and process parameter that impact on the drug substance of the CQAs. So brief description should be there. Brief description of the development of any design spaces should be mentioned. The reason for each significant change should be explained. If suppose initial study we have done some parameter, later on we have made some changes, later on we have done some changes. Each time why you are going to change, on each change what you have achieved should be clearly mentioned. Otherwise we will not understand what is the difference between version 1, what is the difference between version 2, what is the difference between version 3. Because in the product development stages there are n number of trials, trial 1, trial 2, trial 3, trial 4, and changing of the excipient, changing of the process, changing of the drying temperature, changing of the milling, changing of the granular properties, all these things. So because of there are so many versions are created, and out of that best process is designed and fixed. Batch ICH guideline Q11 on development and manufacture of drug substance, chemical entities as well as the biological entities. Information like batch size, scale, site, date, manufacturing, route, process use, and intended purpose should be mentioned. And supporting data for comparative analytical testing. Suppose whatever the product is developed, that should be analytical testing to be done. If that product is already available in market by some other innovator, we have to do the study with that comparative dissolution or comparative analytical study with that innovator product, and we have to prove that our results and that the innovator drug results are both equivalent and same. So we can say that our product is also good as well as similar to the innovator's drug. Manufacturing development studies. Each study and risk assessment should be summarized with level of detail sufficient convey and understanding of the purpose of study. Data collected, how it was analyzed, conclusions, impact of the study on the manufacturing process, further development of the manufacturing process, all should be recorded. Manufacturing process and process control. Process control means whatever control is there, suppose speed of the machine, temperature of the machine, or your timing of the blending. These are all the parameters. So these process parameters should be strictly controlled. Description of the manufacturing process should be provided in the form of flow diagram, sequential procedure narrative. If required, there should be some flow diagram to be mentioned in the process. In the process control, for each step or stage, the process should be indicated with the description. Scaling factors should be included for the manufacturing steps intended to span multiple operational scales when the process step is in skill development. Pharmaceutical process development, impurity generation from API to drug products. It introduced or created early in the API manufacturing process typically have more opportunities to be removed in purification operations like washing, crystallization, isolated intermediates. That impurity generated late in the manufacturing process means when the API, active pharmaceutical drug, is manufactured, that time washing is required, crystallization, isolation. Because of following all these stages, we can remove the maximum impurity, and on the basis of this impurity removal or the minimum impurity, we can have the good results in the finished drug product. The ability of analytical procedures should be detect impurities in the starting material as well as the final drug products. As the product impurity play a hazardous role, because impurity should be always in drug product very less, very minimal, because impurity plays a hazardous role and sometimes it will impact on the product characteristic as well. Developing a control strategy. Control strategy can be developed through a combination of approaches utilizing the traditional approach for some CQS steps or unit operation, or some more enhanced approach. Means we have to define this control strategy with the studies. In a traditional approach to developing manufacturing process, control strategy set points operating ranges for typically set narrowly based on the observed data ensure the consistency of manufacture. Means ranges, operating ranges set narrowly means suppose your heating temperature range is 30 to 50. So based on the over and over multiple trials, it will be set narrow like 40 to 50. So whatever the narrow ranges, it will be observed as a consistency of manufacturing. Suppose if we give the range like 30 degree to 80 degree or 60 degree, what will happen? Some operator will dry on 30 degree, some operator will dry on 60 degree, some operator will dry on 50 degree. So the equivalent drying will not there, and because of the equivalent drying of the drug is not there, the product consistency will not be there and it will be mismatched. The person who will dry accurately will give the good result, but the person will dry on higher side like 60 degree, it may give the bad result as well as the failure of the product. So always the operating range should be very stringent. More emphasis in place assessment sequence and stage of the drug substance. Process later on, once the product is developed and designed, later on we have to establish the process validation or evaluation. Process validation involves the collection and evaluation of data from the process like design stage throughout production that establish the scientific evidence that a process is capable of consistently delivering a qualitative substance. And process validation to be done on protocol based study. Equipment details should be there, process parameter details to be validated, CQA and CPP to be validated, and results recorded, product stability to be monitored. These conditions established by organization like International Council for Harmonization, means ICH guideline, vary by product to product or geographical climatic zone. Stability studies is always performed with respect to geological zones. With common example, 25 degrees centigrade 60% RH is the long-term testing, and 40 degrees centigrade 75% RH is the accelerated stability testing. Means all the zones to be covered for the stability testing. At any zone product go, it should may have stable. Key factors for the stability like temperature and humidity, light study means photostability, whether my product is impacted by the heat or light, that study to be done. Container closure study means bottle pack, carton leaflet, sorry, a blister, strips, small bottle, big bottle, drum, all this type of container closure study to be done. There are the three stability conditions like long term, accelerated and intermediate. Long term means 25 degrees centigrade plus minus 2 degrees centigrade, 60% RH plus minus 5% RH. Okay. Or 30 degree plus minus 2 degrees centigrade, 65% RH plus minus 5% RH, subtropical zones. In long term, there are some temperate zone as well as the subtropical zone. So the long term condition is different, 25 as well as 30. In accelerated, 40 degrees fixed, 40 degree plus minus to 75% RH, it is fixed. In intermediate, again we can say that 30 degree plus minus 2 degree and 65 degree 65% RH plus minus 5% RH. So these are the three conditions for the stability, and basis on this stability condition we are performing the stability studies. There are some of the ICH stability zones like zone 1, zone 2, 3, 4, zone 4B. Zone 1A means temperate zone, zone 2 means Mediterranean subtropical, zone 3 means hot dry zone, zone 4 means hot and humid tropical zone, zone 4B means Asian zone. Okay, now zone 1 means 21 degree, zone 2 means 25 degree, zone 3 means 30 degree, zone 4 means 30 degree, zone 4B means 30 degree plus minus 2, but here the humidity will change. If you see 30 degree, there is zone 3 35% RH, but in zone 4 30 degree 65% RH, zone 4B 30 degree but 75% RH. So accordingly RH condition will be changed with respect to the zone. Same here, accelerated and intermediate stability testing. If you see accelerated 40 degree 75% RH, accelerated 25 degree 60% RH, accelerated frozen 5 degree no humidity, and intermediate 30 degree 65% RH. But it should be done for 6 months. Accelerated and intermediate study to be done for 6 months, long term to be done on 12 months at least. Pharmaceutical process development, with all the process stages and evaluation, product is developed. At last, finally, the product is developed. And once validation and stability of the product is done, this product is ready for the next level, that is the commercial manufacturing. Then this product will be transferred to the manufacturing site called as technology transfer. And all technology like manufacturing process, formula, analytical method validation, stability, validation, photostability, all this technology completely transferred to the manufacturing site for the next commercial, and further commercial to be started. So in this way we are starting from A to Z, from product development, scale up to last commercial manufacturing. We have seen here from basic to commercial stages. So in this way pharmaceutical process development is happened. Hope you like this. If you have any comments, you can put into the remark or connect us, or if you like, kindly subscribe us so that we can give you a next video with a new topic. Okay, thank you.